begins in 5 minutes. Attention. Do not leave any luggage unattended. Unattended luggage will be removed and destroyed. Attention, ladies and gentlemen. This is an announcement from Planet X31. Please take your seats.
One small step for man, one giant leap for mankind. Columbia, this is Houston, may I let over?
This is live to test the reactions of troops to atom bombs used in support of an attack.
1, 0, 0, 5, 2. Lower right.
Greetings, planet Earth, and welcome to the Science and Cocktails. Science and Cocktails is an initiative taken in cooperation with the Niels Bohr Institute, and its purpose is to bring first-class scientific information directly to you, Earthlings, without interference of any other sort, type of intermediate media. Its aim is to make the flow of thoughts and the flow of sensorial perception as smooth as the flow of a cocktail mixture. Tonight's teleportation will be guided by Katherine Richardson, Professor and leader of the Sustainability Science Center at the University of Copenhagen. Her lecture will be about the Galathea expedition, a quest taken by only a few who sought out to search for answers for the survival of mankind in the remote locations of our own planet. Our focus of tonight will be on the answers they found during this quest.
Ladies and gentlemen, the words of Katherine Richardson will be followed by a performance based on hardcore Arduino technology and a barrel organ machine by the artists and magicians Galathea Muller and Sophie Yarth. Let's give a big applause for Katherine Richardson.
I thought I tried just about everything in this branch, but boy, was I wrong. Thank you very much for that interesting introduction, and thank you very much for inviting me here tonight. I really think it's now that we should be communicating Galathea and not however many years ago it was, 2006, 2007, where the whole idea was it was about communicating research and communicating research results. And the last time anybody was interested in my research results was when we were sailing into the harbor on the last day. Well, it was the day before. We came in on Tuesday, and on the Sunday we all sat in the cafeteria with the media experts on board, and they put microphones up in your face and said, "And what did you find out?" I was going home with over 10,000 samples. I didn't know what I'd found out. Now I've got a pretty good picture. At least I found some things, although I will be working on these data probably until I drop. And so there are lots of good questions I can still ask the data, but now at least we're in a position to be able to tell you what we got out of it, or what we expect to, what we have gotten so far, and the kinds of things we may get in the future. A little background on this. Well, of course, Denmark has had Galathea expeditions before. And there was one, number one was in 1845 to 1847, and the second one was in the mid-50s, or the beginning of the 50s. And I actually know some people who have been on that cruise, the Galathea 2, and I have to admit, I kind of laughed at them, because 50 years after the cruise, they were still talking about the cruise, they were still living the cruise, they were still looking at the data, it was still filled this much. And I totally understand where they came from. And when we came back from on the last leg and came into Copenhagen, and there were all those people waiting for the ship to come in, and we had a deal that we could bring our families on board to see the ship if we could go down and find them in the group there, or all the people that were there. So I came down to find my family, doing this sort of, a little bit to the right, a little bit to the left on the telephone to get them over to me. And the first person I met was actually Torben Wolf, who had been on the Galathea 2 expedition. Very old man at that point, he still is, hasn't gotten any younger, but he was waving a Dannebrog, a little paper Dannebrog, and he gave me a huge hug, and he gave me that flag. And I've saved that flag, and I intend to give it to the people going on the next Galathea expedition. We were on a naval ship. The reason that we were on a naval ship instead of the excellent research ship that Denmark has, the Dana, is that it was decided that this particular cruise wasn't really only about science. Science was kind of a good excuse to have it. What it really was about was trying to engage young people in natural science, and therefore we needed to be able to have students on board, we needed to have media folk on board, and so on. And the research ship just simply wasn't big enough for both a crew, scientists, students, and media. So they refitted a naval warship, and in fact we went all the way around the world with a cannon. The cannon was what came first every time. We did try and get the cannon removed to no avail. So they made this ship into a research ship just for the purpose of this, highly, all the modern mod cons, as it were, of a research ship. And as soon as we came back, they paid all that money to take it all off again and rebuild the ship back into a warship. So there were two focuses, or two foci. One was actually doing science and understanding what the ocean was all about. Big difference, though, from Galathea 2. And I think maybe in some ways the media didn't understand that difference. So the messages that got out to people weren't really what it was all about. Because in Galathea 2, what people were really doing was trying to find out what was living in the very deepest part of the ocean. And, you know, they kept pulling up, oh, look, here's a new fish we've never seen before, any formalin and into Zoological Museum, and there it is today. We've come farther than that now. Jacques Cousteau and the BBC and all these wonderful discovery films and so on have brought us all out and seen these fantastic animals. So the focus now wasn't on discovering, although we got lots and lots and lots of new species, don't misunderstand me, but the focus was really on understanding how the ocean works rather than what might be living in it. So that meant that a lot of our things we were working on were actually some of the really small things, and they're difficult to communicate. And we didn't really have a focus on getting the right stories out, I think. The other thing that people aren't aware of is that there was also a culture and a history aspect to Galathea, and the cruise plan was actually made with reference to where Denmark has a historical tradition. I mean, the Virgin Islands, for example, where Denmark obviously has a history. Unfortunately, in the last minute, since this was all timed very cleverly together with the Mohammed drawings, we had to change a lot of the routes so that we didn't go through some areas of the world where Denmark does have a history. And there were projects working on land as well. Now, what was the route itself? The route started in Copenhagen and ended obviously in Copenhagen. It lasted 8.5 months. There were I should say that I was not on for 8.5 months. My project was the largest project. It was on for 8.5 months, and 2 of the people that were in my project were on all the way around. They're the only 2 on Galathea. They went on when we started, came off when we came home, and it took them about 2 years to be normal again. But they had a really, really wonderful time. It wasn't the ship they had a hard time adapting to. It was coming home. I started out by taking the first leg to make sure that everything worked, and then I couldn't get a leave of absence from the home front for quite that long, so I stayed at home until the ship came here. And when the ship came here, I came to Hobart, and in Sydney there were two military crews, and the first one went from Denmark to Sydney, and the second one from Sydney and back to Denmark. And so I came on in Hobart because there was a civilian cruise leader for each leg. It was divided into 23 different legs. You can see them drawn up here. And I was the cruise leader from Sydney to Gizo, which meant that I would be I was responsible for all the civilians on board. There are 50 military people, 15 civilians, the civilians including scientists, media people, and students. And you have the captain of the ship, who has a responsibility for the ship as a whole, the safety of the ship as a whole, and also for the military personnel. And then you have the chief scientist, who's responsible for the 50 civilians on board. And so the chief scientist actually sits at the captain's table, and it's only the chief scientist and the captain, and it's a little... Anyway, it got a little tricky, actually. I'm going to show you some pictures now in a minute to give you a feeling, to get you on board the ship. And this leg where I was the cruise leader actually covered Christmas and New Year as well. So we had Christmas there. And you have to understand, a military ship is a very, very hierarchical and structured machine. And you go into, if you're the cruise leader, you go into the leadership of a Danish warship. And that means you have a meeting every morning at 8 o'clock with the chief of this and the chief of that and the chief of the other thing. And you sit around the table and decide the plan for the day and all that sort of stuff. And you know, I kind of I kind of asked quietly beforehand, is there a special place I'm supposed to sit, and is there a special time I'm supposed to say something? Oh, no, it's completely open. God help you if you sit the wrong place and say anything at the wrong time. But Christmas was on this leg, and there was a big problem because there are 4 places to eat on that kind of a ship. There's the blue room, which is where the captain and the chief scientist sit. Then there's the officer's mess. Then there's the sergeant's mess, and then there's the cafeteria. And you don't eat at other people's messes unless you're invited. So the captain and I had a major problem. If nobody invited us for Christmas dinner, we were going to have to eat alone. And nobody was really sure they wanted the captain for Christmas dinner. But finally, the officers did relent, and we had Christmas with them. Anyway, I went from Sydney to Gizo to Christchurch, then I went back to Denmark, then I flew out to the Virgin Islands, went to Boston, and then flew back sailed back to Denmark. So I was on for 3 months, all in all. As I say, this little leg here, I was the chief scientist, and I want to start this off by showing a cavalcade of pictures that were put together by the engine room. Somebody asked me earlier this evening, and what were you doing all the time? You can't do research all the time. Well, actually, a scientist probably can't do research all the time, but if you're only there to make the engine work, you can't just make the engine work all the time. The whole idea is it's supposed to work. So you have some time on your hands. So they were very good about making a picture for every cruise leg. They made a cavalcade of pictures that gives a sort of idea of what life was on board was like. And remember, when seeing these, that this is all about the changing of a military personnel, the retirement of the captain who was there before, so you'll see ministers and so on. It's unpacking everything for the next 6 months. It's Christmas, and it's a lot of work, but you'll see in a minute. Whoops, I hope. That was before he lost weight.
How do I get through a night without you? If I had to live without you, what kind of life would that be? Oh, I need you in my arms, need you to hold. You're my world, my heart, my soul. If you ever leave, well, baby, you would take away everything good in my life.
The Danes weren't real good at the sun.
And tell me now, how do I live without you? I want to know. How do I breathe without you? If you ever go, how do I ever?
Lots of interest from the media when the samples come up. We were working out in these containers here. Those are Christmas carols being sung at 11 o'clock every morning.
Baby,
Who says science is boring? I should probably say that that town of Gizo that we were at the end here was totally destroyed by a tsunami before we made it back to Copenhagen. So that was it was really, yeah, it was very moving to have been so close to those people, and we actually had a huge collection of money and tried to support them there. But let me tell you a little bit about the project that I actually was responsible for. It was the largest project. It was called the Carbon Cycle from north to south along the Galathea route. And basically what we were trying to do is to understand the carbon cycling in the surface of the water. And I'll explain why we're interested in that before we talk about it. I should probably give a little background that says that, it says that a new discipline has been emerging over the last couple of decades called Earth System Science. It's not about understanding individual processes, it's about understanding interactions on the whole Earth and how it all works together. And basically the message is that we have a system that functions as a single system where ocean, atmosphere, land, and people all operate to make the system be what it is. And given the fact that the ocean covers 71% of the Earth's surface, it stands to reason that the ocean is incredibly important in how this system works. And in fact, we are completely dependent on the ocean for the environment that we have here. We would not be here now if there wasn't an ocean. I should also remind you maybe that life started in the ocean. So it's been very important to the way this planet has developed. It's very important to the way the ocean, or the way the Earth is now. I'm going to take a little aside here and tell you about not Galathea research, but one of the reasons why I am so committed to trying to understand how the ocean works. This is a study that I did together with a number of different scientists from around the world. This was in Nature in 2009. Basically what we did is we said, okay, this business about being a natural scientist is really exciting. You collect data, and 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 then you look back and you can say how things developed. And you can do this as an environmental scientist. You can do this for decades. And when you get enough data in, then you say, oh, yes, well, yes, we made a pig's ear of that. We never should have done that, should we? Could we, with our knowledge of how the Earth system works, could we identify places that we don't want to go? Things that we shouldn't do to the Earth system? And our argument went as follows. We know we know from history that let's start another place. You all know that if you have a lake, a freshwater lake, and you pump in nutrients, then the water becomes cloudy and you get little plants up at the surface and all the plants at the bottom die. Now take away the nutrients. Do the plants in the bottom come back? No. Where should they come from? They may in decades when birds have brought them in, but basically there's a new state, a new semi-stable state that the systems come into. Could this happen to the Earth? Well, indeed, we know it has happened to the Earth. This is, what you see on this axis is a proxy for temperature, and it's a stable isotope, oxygen isotope, and these represent temperature changes over time. We're looking at the last 100,000 years. These data ultimately came from the ice core work that's done at the University of Copenhagen. And these big differences that you see here, those are changes of like 10 or 12 degrees centigrade over a decade. That's huge. So we can say that over the last 100,000 years, our planet's been on a pretty rocky road. And our species, in its present form, showed up sort of over there somewhere, about 250,000 years ago. That means that some of our ancestors survived those climate changes. How did they do it? We can learn from them. They did it in two ways. One, they died. We know from genetic analyses that we were down to relatively few of our species, tens of thousands of reproducing individuals. The other thing they did is when the water got up to here, they moved over there where it was dry. And you could do that when there were tens of thousands of reproducing individuals. We're 7 billion today. We'll be 9 billion by 2050. So that's a challenge. But what's more interesting about this picture is this. the Holocene. This is the last 12,000 years. Look how stable climate's been there. And it actually turns out that that's the period when our species really took off. We'd been there for thousands and thousands of years, but it's only in this period here where we really took off. Why did we take off? Well, we took off largely because we learned to do agriculture. Why did we learn to do agriculture? Well, a lot of people think, a lot of anthropologists believe, that it made a huge difference that you weren't running around all the time to keep your feet dry, but could sit down in one place and watch and see those seeds that went down and came up, and you get this idea, this predictability in the system you can actually use. So our argument went, listen, this is the only period, this is the only type of Earth that we know humans can thrive in. They can survive here, and they can probably survive in anything else, but we don't know if they can thrive in anything else than that. So the precautionary principle says, let's not change it. If it ain't broke, don't fix it. So the first thing you can say is, well, why is it so stable? Well, one of the reasons it's been, or the major reason it's been so stable, is the fact that the Earth's orbit around the sun is particularly circular, has been particularly circular in this period. And so you can say, oh, well, that could change tomorrow, so I'm not going to worry about it. But there are actually people that devote their lives to trying to understand how the Earth's orbit is going to change through time. And those people tell us they expect this circular bit to continue for something like at least 15,000 years. That's good enough for me. So we don't believe that anything natural in the system is going to change that. The only thing that could change it is if we start changing parameters that could push it out of this system. So we went in and had a look at what really changed, or what major changes were occurring in the planet as a whole. And we found 9, and we actually, for 7 of them, not for the aerosol loading and not for the chemical pollution, because we didn't think there was enough historical data to do the analyses. For 7 of them, we proposed limits, boundaries, where you greatly increased the risk of pushing the Earth into another state if you pass these boundaries. And what's really interesting about that is you can then go in and say, okay, listen, we take our 9 boundaries. Remember, there were 2 of them, this one and this one that we couldn't really do anything about. But we can say, here's the boundary. And the boundary could be a little bit in or a little bit out. There's nothing magical about this. But we're proposing those as boundaries. And let's call that the supply. Those are supplies of Earth resources or services that we can use. And then we can look at demand. And here's the human demand for those resources before the Industrial Revolution. And then we move it out into the Green Revolution of the 1950s, and you can see we're pushing agricultural land use, we're pushing climate change, we're pushing the nitrogen flow. And as we go through time until we get to the present day, we see that we are pushing our way out against these. Now, for 3 of them, we arrive at the conclusion, for climate change, nitrogen flow, and biodiversity loss, we arrive at the conclusion that we're on the wrong side of the, I'm not doing this, on the wrong side of the boundary. But that doesn't necessarily mean that everything is going to collapse. I mean, probably there are people here who have been smokers. When you smoke, you run a risk of having smoking or getting a smoking-related disease. When you quit, your risk becomes almost as small as one that's never smoked. Likewise, if your blood pressure is 120 over 80, the doctor will probably try and bring it down, not because it's a guarantee that something bad is going to happen, but the risk is greater there. So what we're saying is that the risk is very great because we're out on these, but this isn't going to go away because the freshwater consumption, agricultural land use, the ozone depletion. We've got the phosphorus use. So we've got this. We're in a position. We're in a really, really wonderful position, actually. There's no reason to be negative about this. In 1987, in the Brundtland Commission, it was admitted that sustainability was not just a question of economic sustainability, it was also environmental and social sustainability. But we didn't know what environmental sustainability is. Now we know. Now the science is telling us it's supply and demand, and we're getting to the point where we can quantify the supply. And that gives us a fantastic power to be able to define some limits within which we can develop and grow our societies. But let's go back to Galathea, which is also, this climate thing is also one of the planetary boundaries that we found. This is fantastic data. This is the most important data. This is the data that made people sit up in 1999 when they came and said, oh, people have been talking about this climate thing for years. I mean, Radius in Sweden in 1896 said, hey, you know, guys, we're burning this CO2. The CO2 is, and he estimated how much warmer the planet would become when you put CO2 out there. You know, even though he didn't have a computer, he got pretty darn close to exactly what we're saying today. So this is nothing new. But so people knew about it, but nobody had any evidence it was really happening. And then in 1999, a Russian and a French research team went to the Antarctic, and they developed this wonderful curve here. It's now been extended back, so it goes to 870,000 years ago. This is just 450,000 years. And what it shows is the CO2 concentration here, the methane concentration here over the last 40,000 years. I told you not too long ago that our species in its present form has only been here for 200 or 250,000, so nobody was measuring at that point. But there are bubbles in the ice, and the bubbles in the ice contain the gases that there were in the atmosphere at the time when the ice was formed. So when you take an ice core up, you can analyze the bubbles in the ice and find out what the CO2 concentration was. And you can see that CO2 has gone down and up and down and up and down and up and down. Those are ice ages that are coming and going. I really don't like this. That are coming and going. But what is worth noting here is that when it gets down to about 180 parts per million, it is, whoops, it's time to go up again. And when it gets up to around 280, it says, now down again. So until very recently, and now we know that this is at least the last 870 or 80,000 years, the CO2 has been varying between 180 and 280 parts per million. Now, we're up at 293. And we know, this is not a guess, we know that that is largely the result of the burning of fossil fuels. Now, how do we know that? Well, it happens that when you burn fossil fuels, I mean, fossil fuels were formed a really long time ago. So their carbon signature, their stable isotope carbon signature is different than the CO2 that we're producing from trees today, for example, or from breathing out today. So we can actually say we know that carbon originated from something very, very old that was combusted, and the step to the oil and gas isn't very far. We also know that at the rate that we're letting it into the atmosphere, we're coming up here, by 2100 we could be at 700 or 1000 ppm. Now it's clear that we humans have never experienced anything like this before. This is entirely new. What's also clear is that the ocean has been taking up, we talk a lot about the CO2 we have in the atmosphere, but the ocean has been taking up a lot of that CO2. Of course it has. 2/3 of the ocean are covered by ocean, is covered by ocean, which is in direct contact with the atmosphere. Now, hang on to your hats and glasses, because I'm going to take you all the way back to 5th grade chemistry now. What happens when you increase the partial pressure of a gas on one side of a membrane? What happens to the partial pressure of the gas on the other side of the membrane? It increases. So we get extra CO2 coming into the ocean, and some of it actually stays there, and we'll get back to that in a moment. But the point is, the ocean has taken up between 1/3 and 1/2 of the anthropogenic emissions of CO2 since the Industrial Revolution. That means without the ocean, we'd be in a mellow S in terms of climate at the moment. So where and how does this uptake occur? Well, this all looks very complicated. Don't worry, we'll get through it easily, I'm sure. This is the carbon cycle in the 80s. It doesn't matter. The idea is very much the same. As I say, we focus. The red numbers, that's where there is increases of carbon or CO2. And we focus all the time on what's happening up in the atmosphere. And we see that number there. But what we don't ever talk about is the fact that we've got a red number here and a red number here. Now, my research is all about this red number, and we're going to talk about that, but I'm going to give you a little hint about what's happening here, too, even though it's not my research, because I think it's an important story. But first of all, let me remind you that if you took all of the carbon that you have in the ocean here, not the stuff in the sediment, but all of the stuff in the ocean itself, and added it up, you'd find out there's about 50 times more than there is in the atmosphere. And the atmosphere is in contact with the ocean over 2/3 of the planet. So you tell me how you're going to predict what's going to happen to CO2 in the atmosphere if you don't know what's happening to CO2 in the ocean. But let's take a quick look at what's happening here. This, as I say, is the old business of you've got the partial pressure is greater here, so you get more CO2 coming into the surface. Well, what happens when you put acid or CO2 into water? You get acid. That's why a lot of teenage kids have acid marks on their teeth from having too much soda water. That's why even Danish water is bad for your teeth, because you've made carbonic acid, and carbonic acid reduces the pH. It increases the acidity. We have lots of proxies where we can measure the change in acidity or the change in pH, which means the change in acidity of the ocean over the last 23 million years. And you can see that the pH that has never been has never been lower. That means to say it has not been more acidic, the ocean, than it is today over the last 23 million years. We can also calculate, if we keep putting CO2 out into the atmosphere, how the acidity is going to increase. And you look at this pH and say, well, who cares? It's still higher than my shampoo. Yes, this is true. However, there are lots of organisms in the ocean that make chalk. There are fish gills, there are lobsters and clams and all sorts of corals, all sorts of organisms in the ocean that make chalk. If you go home and take some vinegar and put some baking soda and put the vinegar on the baking soda, you'll figure out what happens to chalk when it acidifies. It dissolves. Now, this is important because we have some of these little organisms, for example, that are exceedingly important in terms of transferring carbon to the bottom of the ocean and making Mons Klint, for example, is made of these kinds of organisms. Well, they are completely made up of chalk. But other organisms that you may know better are corals. And corals have a particularly, their structure, their carbonate structure, the crystalline structure of their carbonate is exceedingly sensitive to changing pH. And here you can see, it's really neat because you can calculate, you can sit at your desk and calculate, knowing the temperature and the concentration of CO2 in the atmosphere, you can calculate the conditions in the ocean and whether or not the chemical conditions are conducive to corals growing and making their skeletons big. Now, this is before the Industrial Revolution. This is before we started changing the CO2 concentration in the atmosphere. And the dark green areas are where the chemistry is ideal. Chemical conditions are ideal for making coral reefs. The green ones are okay. The brown and the red, we don't expect to find them. Now, all these little black dots, that's where Mother Nature put coral reefs. Pretty smart, huh? What's it look like today? Well, the dark green areas are shrinking. Fortunately, most of our coral reefs are still found in the light green areas. What's it going to look like in the future? Well, we don't need to go to 2100. Already by 2065, there will be no regions in the Earth's oceans at all where the conditions are right for making coral skeletons. Now, that doesn't mean they're all going to go extinct. Some live happily without their skeletons and so on. Does mean you're going to see major changes in coral reefs and their ecology and all that sort of stuff. It's a pretty major change to make in the ocean. Now, climate is a really, really difficult thing to communicate. It is about the Earth's orbit. It is about sunspots. It is about how much water there is in the atmosphere. It is about greenhouse gases. It is about land use change. It's really hard to communicate in those sort of 30-second sound bites that you get on Danish radio. This is a much, much simpler story to communicate. What's causing? There are only 3 things that can cause this acidification of the ocean. CO2 in the atmosphere, CO2 in the atmosphere and CO2 in the atmosphere. And that makes it dead easy to communicate. The problem is that nobody really cares. Okay, now let's go to this part down here. This is what my research is really all about. There are both physical and biological reasons why you can get carbon down to the bottom of the ocean. The physical ones we know quite a bit about. It is, for example, when you make icebergs up in the Greenland Sea or the Labrador Sea. And when you make icebergs, what happens is, as the fresh water freezes at a lower or a higher temperature than salt water. So when you've got salt water and you freeze it, you basically freeze fresh water and push the salt out of it. So you get some salty water around this ice, which is very, very cold, and when it's salty and very, very cold, it's very dense, and when it's very dense, it sinks, and when it sinks, it goes to the bottom. And cold water actually can have more bubbles, more CO2 in it than warm water. And if you don't believe me, take a beer, let it warm, and then get a cold one, and then see how many bubbles there are in the two of them. And so you have this water which has lots of CO2 in it. It sinks to the bottom, and when it gets down here, it can stay for 1,000 years. So it's no longer in contact with the atmosphere. You don't get it back out again. So this is a really nice place to have it at the moment when we're dealing with it. Those are some of the physical reasons, but I'm interested in the biological reasons. Now, what biological reasons could they be? Well, here we have the tiny little plants of the ocean. And I have to say, I did not get into this business because I wanted to be a climate scientist. I got into this business because I fell in love with these guys. You think it's crazy, but I fell in love with them. They really are absolutely fantastic. You will read in any textbook, it's just a little black box, phytoplankton. They're small. We can't see them. But that doesn't mean they don't have a fantastic story in and of themselves. And in fact, the size difference, even though they're all very small, the size difference between the largest, these are just from the North Sea here, the largest, this is a Cuscinodiscus, and the smallest, which is basically these little things you can't even see, the size difference is actually greater than the volume difference between a mouse and an elephant. How many people think that mice and elephants do the same thing in the food web? Good. The point is, it makes a huge difference whether you have large or small phytoplankton for how the system works. But these are the plants. And there's just as much photosynthesis going on in the ocean as there is on land. They do their photosynthesis, and just like we say, plant a tree, you'll save the world from climate change. Well, you get CO2 coming in here. Most of it gets respired out again, but some part of it falls down here. And when it comes down here, it stays for, as I say, up to 1,000 years. And so it's away from the surface. It's not influencing us in terms of climate change. So you've got this carbon, which basically is just on a short visit. But this stuff really makes a difference in terms of trying to understand climate change. And that's what my research is all about. What biological factors are important and how much do they add? So basically, we've got CO2 in the atmosphere. It gets in by diffusion to the surface ocean. You have physical processes that can bring it down here, and then you have these biological processes where you have these phytoplankton or these small plants that take up the carbon, and when that part of it that sinks can go down to the bottom. And normally, there are a couple of ways we can try and figure out how much it is. One way is by putting traps down there and trying to collect the stuff that falls down, but I have to tell you, it's a very big ocean. And the other problem is you always have to come back and take the trap up. And time was our worst enemy in the Galathea expedition because the ship always had to sail somewhere else. So it actually ended up that we had to model the carbon falling down once we had the processes, control over the processes. What kind of processes were we actually looking at? Well, we took lots of CTDs where you put this is a conductivity depth, a temperature depth recorder. It measures profiles of lots of different things, oxygen and plant material and salt and temperature and so on through the water column. And then we had bottles that we brought samples up and measured all sorts of stuff, everything from enzymes and genetic stuff to what species were actually there. We were measuring the rates of photosynthesis. We were looking at the small small fish to see what they were doing isotope analysis and so on. So basically we were trying to quantify the movement of everything through the system, and then we were also measuring the exchange, how much CO2 was coming out of the ocean and how much was going back in again. And we have ended up with a totally unique data set on the distribution, the biodiversity of these organisms. And lots of really exciting stuff is coming out of it. And what's that so far? Well, first of all, changing ocean conditions are changing this diversity and thereby changing carbon and nitrogen cycling. What we found out, and this was a surprise for everyone, is that the size of the phytoplankton that you have is a function of temperature. It's also a function of nutrient availability. Everybody always knew that. What we didn't know, because most people just do this study in their own backyard where the temperature variation is from 6 to 10 degrees or whatever, is that temperature also is a factor here. And we were able to do sampling from below 0 degrees to over 30 degrees. And this picture shows you the median size of the phytoplankton when it's 2 to 6 degrees, the medium size from 6 to 14, and from 14 to 30. So a warmer ocean will mean smaller phytoplankton. Now, why should we care that they're getting smaller? Well, Clinton, I guess, was it Clinton that in some, he was giving a State of the Union or something, he had a big sign up in his office and it says, "It's the economy, stupid." But when it comes to phytoplankton, it's the size, stupid, because the smaller ones here can only be eaten by very small animals that then get eaten by slightly larger animals that again might get eaten by slightly larger animals and then might get up to fish larvae. Whereas the larger ones can either be eaten directly by fish larvae or by the large ones and then fish larvae. What that means is there's only 2 trophic levels to get it up to us, whereas if it's small ones, you can have 4, 5, or 6 trophic levels. And many of you will recall that every time you move up a trophic level, you lose about 90% of the energy. So if you have a lot of small phytoplankton, you have less energy coming up to the top of the food web. So small cells will change the food web structure, and it will lead to having smaller zooplankton. It will lead to having more links in the chain. And it also, large cells sink faster, so it's easier for the large cells to get to the bottom than it is the small cells. And indeed, if you take our data, again, this is new data from Galathea, which shows that this is the reproduction of the larger zooplankton. And it shows that at the low temperature, they're very reproductive, there's more of them, and when you get to the higher temperatures, there's less. This isn't a temperature thing, it's the fact that there are smaller things to eat here and larger things to eat there. So this is a universal relationship. Again, not something that people have been able to look at around the whole world until we had Galathea. The other thing is that we can say that cell size is related to the amount of CO2 coming from the atmosphere into the ocean. This is just data from the North Atlantic where we had a lot of seasonal data from Galathea. And you can see the larger the organisms, the larger the phytoplankton, the more CO2 is going into the ocean rather than coming out of the ocean. The other thing we found out was that it doesn't just matter how much nutrient is available. The ratios between nutrients are hugely important for the size of the phytoplankton. This is what we call the Redfield ratio. It's a sort of the common phytoplankton, the common seawater ratio of nutrients if you get away from a coast which is affected by our runoff from land. And you can see these are the large cells. When they come up here, they're large cells, and when you're down here, they're small cells. And you right around this point, which is just like the open ocean, you get the large cells. And if you change that ratio, you get smaller cells. Now, even though we were all the way out in the middle of the world's ocean, this is something which is very relevant. This is, again, putting this in boxes, and we can see that right around the Redfield ratio, the cells are larger. But even though we're all the way out there, what we found here is exceedingly relevant for understanding eutrophication, or iltsvind, the oxygen depletion events that we have in the Danish waters. Because the ratio of nutrients is what we really change. When we run off a lot of nitrogen from the farming land and so on, we actually are changing ratios of nutrients, and we're probably getting very small cells rather than large cells and changing whole food webs. I thought I'd take a little diversion and tell you about some newer studies also about the inner Danish waters. This is using data, lots of data from 6 different stations here. And the point of this data is that we do actually find a relationship between total primary production and land-based nutrient loading. The nutrient coming from Denmark in the wintertime does control how much primary production is out there. We sort of expected that, but what we didn't expect was the fact that it turns out that when we look at our total primary production, we have about 17% of it occurring in the bottom water itself. We've always assumed that it was all happening at the top, but no, there's about 17% happening in the bottom layer, below the surface mixed layer. And what we found out was that the amount of production happening in the bottom layer is actually also a function of how much nutrient is coming off from the Danish land. What that means is the greater the nutrient runoff, the less production happening in the bottom water. Who cares? Well, actually, it's a good reason to care. Because when you do photosynthesis, you produce oxygen. So when you have production happening in the bottom water, you're actually producing oxygen there. And one of the reasons why we may be having oxygen depletion in Danish waters is because we've moved everything upstairs. So we're not using that oxygen anymore. But even more importantly, it turns out, this is looking at size again, where these are the very small organisms, these are the big organisms, and you can see that right down here in this layer, in the bottom layer, it's big organisms and it's small organisms here. So we've changed whole food webs by putting nutrients in. And we've always communicated oxygen depletion and the problem in the Danish waters as being a total production of organic material leading to oxygen depletion. No, it's a complete change in the ecosystem we have out there. We just hadn't noticed it. Another thing we did in Galathea was to quantify what temperature means for how much of the material that will fall down to the bottom. Because you all have a refrigerator. Why do you have a refrigerator? Refrigerator slows down bacterial degradation. And as this material that the plants just picked up, this photosynthesis they did, as they sink through the water column, the bacteria are working away at them. And they won't make it to the bottom if it's really warm, because the bacteria will degrade them. Now, that's not rocket science. Everybody knew it was going to happen, but nobody had a number on how quickly it was happening. What we did is we took samples and we incubated them at 5 degrees above where we were. We took them at different depths. We incubated them at 5 degrees above and 5 degrees below, and we actually measured over a long period of time how much oxygen was used, how much bacterial activity was going on, and we ended up being able to quantify that if you increase the temperature by 10 degrees, they would dissolve the bacteria would break them down 2.4 times faster. And for the dissolved organic carbon, it was 3.8 times faster. And what that means is that we can go in and actually say, okay, if we say that the present we look at how much carbon's going down at the present, we can say at the last glacial maximum, there was more, and in the future, there will be this much less. If we say the future is 2 degrees warmer. What all this translates to is that if you increase the surface ocean temperature by about 2 degrees, it will increase the CO2 concentration in the atmosphere by about 13 ppm. I mean, we can go in and quantify the changes that we can expect from just being warmer and the changing in bacterial activity, which is hugely important in predicting what's going to happen in the future. Another thing we learned is that the satellite methods we have for predicting primary production don't work. Here we have, these are from all our data on the Galathea, where these are the estimates we made of primary production. These are the satellite ones, and this is on a log-log scale, which means basically the satellite says that there is about through 400 milligrams of carbon per day being fixed here, and our measurements say that it's anything between about 20 and 1,000. And to be honest, I could sit home at my lab and point to any point on the map in the world, I think, and say, oh, I think it's going to be between this and this. So I don't think we're doing very well on these satellite measurements. And I have to say, you could say that it's either because the algorithm's wrong, or it's, maybe it's the way it's measuring the surface temperature or whatever. Because we have this wonderful data set, we could go in and say, no, we'll put in the actual data so we don't have to worry. We'll check it. It's the numbers. It's the algorithm that's wrong. And we're working on trying to make it better with the knowledge that we've gotten from Galathea. Finally, we have some regional stories as well. I've been talking at the global level because I think that's the most important thing about Galathea, is what happened at the global level. And what happened at the global level is what it was all about. It's what I think they should have gotten all of Galathea to be, is to look at the global level. But lots of good stories came up at the local level as well, and this is one that maybe that's what the problem of the thing. Anyway, this is one that came up as we were in the Sargasso Sea. We were in the Sargasso Sea on a, well, that wasn't the one I wanted to show. Okay, never mind. Oh, I've lost the slide I wanted. But anyway, the point is on the Sargasso Sea that people were looking for eels. And I was doing my own thing quietly in my little lab, and not really, in fact, it was so boring. I was looking at the flow of electron transport in photosystem 2 from water at 130 meters and loving it. And they were out there looking for eel larvae, and every time I heard them out there saying, yes, they're here, yes! I suddenly realized that I actually had high ratios as well. There was no reason why there should be a link, but that got us sort of interested in trying to look at, why would there be a link? And it turned out that here you can see comparing two transects, where these are the eel larvae, and the red and the blue, and the red and the blue, and you can see that there weren't very many eel larvae here. There were lots of eel larvae there. What's the difference? It's actually these bars here which represent how much mixing is going on and when you're bringing nutrients up from the bottom. So we can go in and identify, why is it that there were eel larvae right there? And we can actually then calculate how much more productive these regions were. They were about 10 times more productive right where you found the eel larvae than anywhere else. But what was maybe even more interesting, and we're almost at the end of the road here, but what was more interesting was the fact that everybody's always said the Sargasso Sea is a desert. It's a desert because there's not much photosynthesis going on. Okay, but there's eel larvae. There's lots of things they work very hard to get to the Sargasso Sea to actually eat. There must be something there. And what we're suddenly realizing is this division that we've always had between phytoplankton, the little plants, and zooplankton, the little animals. Because of what we know on land, we think, oh, there's these and there's these. Turns out there's very many that are both. In fact, there's some really amazing stories coming out. These are little phytoplankton that form armies and go in and attack the larger organisms and eat them. This one's a neat one. This is a little animal that eats one and only one kind of phytoplankton. It takes it in. When it takes it in, it says, gotta eat this. But when it gets down to the chloroplast, it says, oh, I don't need to eat the chloroplast or digest the chloroplast because I can use that, the guancon. And so it keeps it and uses it and photosynthesizes with it. It stole it, and it's using it. And something in it says, I can use everything else and digest everything else, but not that. I can use that. It does work for me. But then this guy comes along, and he eats this guy, and then he uses the chloroplast again and moves them out to it. So there's all sorts of fantastic stories where it's both plant and animal going on. And when I look again at my data, that was my Sargasso. That's what I wanted to show you, where we were in the Sargasso Sea. Anyway, when we look at the data here, it turns out that most of the phytoplankton, or most of the plankton we have here, are of the kind that do both. They don't just do photosynthesis, they also eat other organisms. And I'm absolutely convinced that one of the reasons why you get like the eel larvae in the Sargasso Sea is there's much more production going on. There's much more carbon coming into the food web than we'd measured. And I'm planning and hoping to go back to the Sargasso Sea in 2014 and check that hypothesis. Now, this has been rather long. We've come a long way around, but the message I wanted to give you was, yes, in terms of science, Galathea was a huge success. I've only told you about one project, where we are now. We have a lot coming. We have a lot more coming online from our project, but there were lots of other projects. And new species, new processes, it's just overwhelming. It was also a success in the sense that different groups were brought together, different scientists were brought together. There's a network. Many of us now have a network with the journalists that we were together with there. In fact, people who are on the Galathea are like a little club, and when you see each other, you are immediately thrown back to another world, and we use each other very much. But what I think everybody expected Galathea to be was an adventure. And some people were disappointed when they came out there and were communicating back home, and they couldn't find the adventure. And therefore, they started turning their cameras on the scientists, and it got a little personal at times, I have to say. It was an adventure if you just remembered to look the right places. And I'd like to finish with another little clip. Apologies for the music, but that's what they put on. Tak for i aften.
Thank you for the presentation. It's a nice ending to it. So we have time for a few questions. So if anybody from the audience, just raise your hands. Or shout if I cannot see you. There.
I was wondering, you showed the map of